Okay. Hi, everyone. I'm Aysun Urhan. I'm a PhD student in Delft Bioinformatics lab in Netherlands. I'm in my last year finishing my PhD, and I'm going to talk to you about SAP, a tool we developed to predict gene function in bacteria. Um, just a quick recap. We know what we're all here for. We know there's a lot of proteins out there that we've sequenced, but we don't know their function. And especially when the amino acid sequence itself, it has no homologs in the known databases. It's a completely new protein, and we don't really know how to. We need alternative ways to predict the gene function. I've noticed this problem, especially when some of our collaborators, they went out, sequenced some bacteria out in the wild, and there were new novel species. They discovered new species, and they had new genes that we had absolutely no idea. There were no known homologs in our databases. And this is very interesting because these bacteria in the soil, in human guts, it's where the antimicrobial resistance originates, and then these genes make their way into our hospitals and cause tremendous outbreaks. So we want to stop this before it comes to that step, and that would be ideal. And lately, as we've been borrowing a lot of ideas from natural language processing, just in short, natural language processing, we have these language models, and when we employ them to represent protein sequences instead of the amino acid sequence, we use these protein embedding vectors, and the state of the art transformers, we've seen them being applied in bioinformatics and a lot of different tasks because they can be used in transfer learning. So they are pretty much task agnostic. You can use them out of the box. You can fine tune them to your specific problem. But there aren't that many applications on bacteria. There are very few of them. So I thought, why not we should use them? Why don't we use them on bacteria? Especially because they, like a step above the amino acid sequence, they can provide information about the fold, the domain, and the structure. So I thought to be able to use them on bacteria, we can also exploit what we already knew about bacterial genomes. And that's also, maybe you've seen, if you've been able to catch some of the talks during this session, it's the idea of operons. And just again, in short, if you weren't there in the talks, in bacteria, we have these genes are usually found in clusters. These are clusters that we call operons. These are conserved across several species. And these clusters of genes are usually transcribed together. So they're turned on and off together. And often they function in the same biological pathway, in the same process. So their functions are also related to one another. Intuitively, in our work, we already use this. When we want to understand the function of a protein, we don't just look at it individually. In bacteria, we also look at its surroundings. I thought it would be better to incorporate this within our tool itself. That's why in our tool, we make use of these two ideas. One, using word embedding vectors to represent the protein sequences. And then two, we also use the contextual information of the gene, the operons, to predict gene function. To start off, I needed an operon database. And this is, I think, an ongoing problem because most databases out there, the legacy databases, they're outdated or they're very limited to the bacteria that they're to the content of the bacteria. So, and I wanted something that I could use in my tool I call SAPDB. For that, I made a really simple computational approach. It's very brute force, very naive approach. I just downloaded a whole bunch of bacterial representative genomes. I got the coding sequences from them, I think from Genome Taxonomy database. At that time, there was roughly 50,000 genomes. I just found these conserved regions of 2,000 base pairs, the neighborhood size, and I also had additional filters like the intergenic distance and just usual heuristics that we know from experimentally determined the real operons in bacteria. yeah, and just after applying these filters, next step was to introduce the language models themselves. So instead of the amino acids, I used embedding vectors to represent each operon, and I also transferred Go terms to these operons. So you can imagine our operon database, SAP-DB, is just a list of thousands of bacterial operons, or just conserved regions across several bacteria, the entire bacterial kingdom. And each operon has two features, one for the embedding representation and one for the Go terms. And then it comes to transferring these Go terms to the query proteins that we want to predict their function. There again, I thought I should go for a very simple nearest neighbor approach here, because that's also been quite successful. And what I do here is very similar to what's been done with GoPret Sim, if you're familiar with that from Ross Lab. Because I have this collection of operons, I just find the nearest neighbor operons just using cosine similarity. I retain only the top 1% of the most similar operons. And from those, I just weigh down their Go term frequency, and I transfer those Go terms to the query point, and that becomes the predicted function. I quickly glanced over that part, but I'm hoping that was clear. But so because I really want to show you some of the results at least. The first one is I'll touch on briefly to make it a bit more realistic. There's no straightforward way to benchmark or evaluate our operon database. The thing that made sense to me the most to do this at a large scale was to compare them to ODB. ODB is a collection of experimentally determined operons that they manually put together from literature surveys, from literature studies. So it's very limited in its content and in its size, but it gives you a good idea about the how an operon should look like. I compared them in terms of the aggregate statistics. Let's say here I'm showing you only the results for the number of genes in an operon, but the distribution of the experimental database at the top and at the bottom are predicted database, SAP-DB. They are very similar, and we also see similar results in terms of the distribution in things like operon length or like intergenic distance and things like those things like that. So at aggregate level, they do resemble one another. And I also found that I can reproduce some of the operons individually. I'll show you some results for E. coli. Handpick a nice one because it's a very manual process, but just to give you an idea, in SAP-DB, we could reproduce the operon, but on top of that, I noticed the operons that I predicted, they are bigger. So we also have more contextual information about the operon, where the operon is found on the genome. I thought that was pretty cool. And at the same time, we also have a whole lot of metadata information from the genome taxonomy database. So with our tool, you can not only predict the gene function, but you can also do additional inference on the gene itself, where it's located on the genome, what other species that it's found in. And then the final step was to benchmark the actual prediction tool itself, SAP. We did the standard approach here, just took the SwissProt database and retained only the proteins with experimentally determined annotations. I'll skip over this for the sake of time, but the important bit here is, because it's focused on bacteria, I created these test sets with 5 of the most populated bacterial species on the SwissProt database. And then, because we're most interested in predicting completely unknown new genes, we created these training sets with different levels of homology constraints. So the constraint, the level of the percent identity between the proteins in train and the test set were different. I varied this from 40% to 80%. And that means we had like roughly 30 different experimental data sets to work on. I compared them against baselines and the state-of-the-art DeepGo Plus, and with the usual metrics. And we confirmed that, yes, word embeddings also work and a lot better in representing and giving information about the gene function. It's better than the amino acid sequence itself on bacteria as well. And we also shown that, yes, I'm almost out of time, so I'll just quickly go over. We also shown that our tool, combining these two ideas in our tool, we're better than the baseline, and we're also better than the state-of-the-art DeepGo Plus. And this is consistent across different levels of homology. I'll stop here. I won't go over the conclusions. And so if you want to hear more about it, just stop by during the poster session or have a look at the bioarchive preprint. Yeah, I won't be able to take any. I'm sorry.